So, Mark, first, I want to thank Mark and say that, Wednesday, April 24th, we're going to have a nice event. Everyone's invited. You'll be getting calendar invites too, but I just wanted to let you know now, 5 o'clock, and then we gave Mark the nice room, which is, we have to borrow from our cancer colleagues, on the 6th floor of Benson. We'll have a reception for Mark, so please come by and tell him goodbye. Mark, I'm going to personally thank you now because Mark came 4 years ago, and we had really big plans for what we were going to do with research, and he started in January, and of course we had our first COVID case in March, and then it went all hands on deck, COVID research, and everything that came with, what we dealt with COVID over the next, 3 years. So, but that didn't faze Mark one bit. We did have a lot of, everybody in this room can attest to, and you've all helped solve many of the issues, but we had issues. To be honest, we had a lot of issues in research. Ari was in my office every other week, but Ari went alone. And Mark, you really did bring structure and professionalism and really worked diligently to address a lot of those structural issues we had. And I'll tell you, over the last 4 years, we've seen grant funding double. We've seen, publications increase. We've seen the expertise of the CRCs and the data scientists increase. And so, Mark, that's all to your leadership. So I want to thank you for that, because I think you really have laid a foundation and built a structure that's going to carry us into the future. And it's really perfect timing, especially with bringing on the Xavier Medical School. But he's retiring at the end of the month, and Joey, I caught him on a weak moment when he was not of all his senses, and luckily he agreed to become the interim chief scientific officer. So Joey will kind of oversee and take over for Mark on May 1st, and make sure that all of the momentum, all the things we got going, kind of can keep going. As you know, we did a search and decided on a candidate. So many of you on the search committee, so you guys probably know that Dr. Nekajeka was the candidate that we decided on. We offered her the position. She verbally has accepted the position, but she has a large lab at UT Southwestern with biospecimens and data. And we're having an ongoing conversation with UT Southwestern about how do we move the biospecimens, how do we move the data, the IP, and that kind of stuff. So we're working through it. It's actually been pretty positive. We had some good communication last week between the two organizations, so I think it's moving forward. But it may not be till August or September that she can come. So we should all buy Joey a beer, or at least a good cab, or but Joey, thank you. In all seriousness, thank you for stepping in and doing that. And so I think that's what stability. I mean, Joey knows the ins and outs of Ochsner. He knows the ins and outs of research, and we're spending the next month. Mark can speak to it a little bit when he talks at the end, but we're going to spend the next month making sure that's a really smooth transition, and that we've checked every box. Questions on that? Look, and it's not like Joey has to take over all of Mark's work. I mean, we're spreading things out. Mark had done a lot on the IRB side and the HRPP side. Dr. Efron is completely taken it over, completely off of Dr. Roberts' plate. Ebony is going to pitch in and help on some of our strategy for hiring a primary care researcher and to be able to do more of these outpatient clinical trials. Haywood is going to kind of take over that. I think there's a great team in place of directors. You know, Ansley's there, and Nicole just stepped out for a second, and you know, Johanna in Baton Rouge. And I think there's a great core team of leaders, and of course, Randy has financials pretty locked down. We made big progress. We're talking about residuals and how to share them, which, you know, 5 years ago was just a concept. So I think there's a lot of key people in place, and Mark, you'll be missed, but I have all the confidence in the team and their ability to step up. Mark, say a few words. All right, any other questions on the transition? We'll know it went well if Research Week goes off well. Mark, if we're struggling Research Week, we'll blame you for the handoff issue.
There's no way we'll struggle on Research Week. You guys show up and you're going to be blown away. It's going to go great. We got the right people leading it. So just come in and have fun during that whole week. It'll be great.
Thank you, Mark. Thank you, Dan. Thank you. Thank you, Leo. I'm going to get this right. Try to. So you guys can see better.
Next online, we have Dr. Mark Matrana, who's going to give us a bit of an update of our subcommittee on research and academic culture.
Great. Thanks, Dan. Can you guys hear me?
Yes.
Okay, great. We can go to the next slide. So thanks for having me. This is going to be very brief, but a very quick overview and update of our academic subcommittee, academic culture subcommittee. And so some of you know about the work we were doing. I think the situation, as Leo mentioned, is that, you know, we as an organization are becoming more and more academic, particularly with the new medical school coming online. And I think we all recognize that the compensation models for physician researchers have to evolve just as the organization is evolving. So we can go to the next slide. Let's see, go ahead and click it again, Dan. There we go. There we go. So to this end, our little subcommittee, which consisted of Dr. Mark Roberts, myself, Dan Morin, and Nicole Vilmarette-Pittman, engaged with several leading centers from around the country: Mayo, Cleveland Clinic, Leahy, Geisinger, and Cedars-Sinai. Very different places, but all places that really succeed at research. And we met with really their top research folks: executive dean for research, chief research and academic officer, so on and so forth. And we spent about an hour with each of these guys and picked their brains on a lot of different things about their culture and their structure, but particularly we focused in on models for physician compensation. Go ahead, Dan, and advance for me, please. And so what we found is that they had a lot of different tactics for success, but there were some common themes. So among those, that academics and research were incorporated into clinical care. So there really wasn't an academic silo over here and a research silo, an academic silo over here and an operational silo over here. The operational leaders were involved in research. The research leaders were involved in operations. It was really fully integrated, and each patient was seen as a research patient. We also learned that each of these organizations recognized that scholarly production was integral to clinical achievement, that you couldn't have one without the other. You had to have research and academics if you were going to grow clinically and you want to be successful clinically and really get to that level that Mayo and Cleveland Clinic have achieved. Next. And they also all had very formalized programs in place that provided early support for talented, motivated, early career physicians. And then they adjusted that support based on how those people performed and evolved over time. Next slide, please. And so we propose a pilot. We met with Dr. Larned in oncology and Dr. Zweifler from neurology. Oncology was already creating a plan. We have supported our physicians in this way. And so we kind of took what Dr. Larned was doing, and she called me a few minutes ago, said she was sorry. She couldn't make it to our meeting tonight, but she had an event for one of her kids. And what we're proposing based on our meetings is that we have a pilot of a research track for a subset of new physicians who are interested in research. So this track would basically include some dedicated time. How much would be a little bit up in the air, but maybe 0.1, 0.2 for research, dedicated time for research. It would have specific education for these physicians and support for a fixed period of time, maybe 3 years. And, we give all these people, we're investing in them. We do everything we can to make them successful. And if they are successful in getting external funding, whether it be from grant funding federally or otherwise, philanthropy, clinical trial leadership opportunities, this sort of thing, that we would then continue to support their dedicated time moving forward. And we would adjust it based on how successful they've been. Next slide. And so we propose that we start this with oncology and neurology, one, because oncology sort of doing this on their own already, and moving forward this with neurology. Dr. Zweifler is very interested in supporting this sort of thing. And, our next steps, Zoe actually met with Leo briefly last week about this, but to really meet as a committee, you can go to the next slide, Dan, with this executive team and tell them what we're proposing. And see if everybody else is on the same page with this. We definitely want to get your input as well, and we'd love to present the findings of this pilot, at least the early findings, at subsequent PSRC and PRC meetings. So, any questions? Any feedback?
What kind of support this decision is going to get? Are we
going
to have protect?
Yeah, I think that's still somewhat up in the air, to be honest, and we're still building and thinking about this. But definitely, in addition to dedicated time for this, you want to make sure that they have the research staff to support them, which oncology and neurology have those built in. Good mentors and operational leaders from the department side. Both of those departments have that already built in, and then some mentorship and education from folks who are successful in clinical trial and grant already. That would be the main things. And then as far as formalized education goes, that's still nebulous. I think that's still to be determined.
And HEMONC is certainly the furthest along in this. They have written out actual plans and proposals and whatnot, and they've been doing over time. They use sort of a scoring grid in order to make a certain number of points if you write a paper or if you give a podium presentation or if you mentor a fellow, for example. So they're well ahead. Fast behind them, I think, is neurology, who are quite interested. And I think as we talk about this more, there may be other departments who want to use the present resources that they have and sort of develop that along those lines as well as we go forward. And then we'll see, as we get success with some of them, and we'll sort of find, best practices and optimize it maybe for the system.
Yeah, our academic points grid has evolved over time, and we'd be happy to share it with anybody in oncology. It's one of our value-based goals for the department over the year for a specific number of points, usually 750 points. And we base it off of, like Dan said, publications, and we take into account the impact factor of the journal that the publication is published in. We take into account podium presentations, trial leadership, mentorship, work that's done with fellows, presenting at the fellows conferences and so on and so forth. And we track that for every member of our department, and we present those monthly in a really kind of sophisticated Excel file grid.
I think it's great to have this metric, and we use in our department and probably also academic productivity as part of the value-based unit. We've been doing it for 2 years. But I don't think that addresses sports, We were kind of recognizing the academic culture by having a value-based, but that's not effective time.
Dan, I couldn't hear that quite so well. Could you?
He was saying that he appreciates, and actually in nephrology, they do sort of a similar type of scoring grid.
Yeah.
But that point doesn't necessarily address the issue of the academic physicians having the support that they require. But I would say, Juan Carlos, that I'll partially answer for Mark here, and then give you a moment as well, Mark. So, for example, in the proposal from HEMONC, a clinician researcher would start with some amount of dedicated time for research, and also there would be some resources given to statistical support and research, grant writing, et cetera. Some of which already exists within our research department and is available to you today.
Yeah. I would agree with that, Dan.
Other questions for Mark?
It's nebulous, right? Oh, go ahead.
No, I was just going to say there's going to be more to come, but
Yeah.
certainly if you have.
Absolutely. A little nebulous right now, but more to come. Just want to let you guys know what we're working on. Thank you.
Thank you, Mark. Speaking of Marks, here's another one. So we have, as you know, we're nearing the end of the tenure of Dr. Mark Roberts, who's been our Dean of Research for 4 years. And this is probably the last meeting of the Physician Research Council that Mark will at least be Dean of Research for. So I wanted to give him some time to speak a little bit. Mark, you're on the line. I'll be happy to advance the slides when you want me to.
Thanks, Dan. Good evening, everyone. Just before we get to the holiday events this weekend, I wish everyone happy Easter, peaceful Passover, and the very best. I'll try to talk through a couple of things that I experienced here. Hopefully it'll be amusing and fun. So thanks, Dan. And go ahead and jump to the next slide. And very good. I'm going to actually have 3 chances to talk to various audiences, starting with you tonight. And I wanted to talk about my ideas as a young guy, especially hopefully appealing to the many of you in the room and the more than 30 folks listening in that are early in your career, how you might translate ideas into action around research. And then I want to end with some thoughts around leadership and things you may incorporate into your own roles advancing Ochsner going forward. So I'm going to, in a couple of weeks, speak to our all-staff folks. We have a couple 100 people that join there, the folks that really do all the work, your coordinators and everyone else, and talk a little bit about my personal story. Hope I can inspire them as well, coming up as a kid and how I found my direction. And then lastly, as Leo said, I'd love to see you all. I'll be there in person on Wednesday, the 24th. I'm going to feature a top 10 list of the research contributions over the last 4.5 years. So thanks earlier to you, Leo, for the kind words. And Dan will go on. For me, when I began, and it's easy, I start often with, I don't know. And the only way really to learn something new is to give up on thinking you already know all the answers. And next slide, please. So, some very clever people in this world have invented things and come away with better ideas by saying, I don't know now, but I will know later. Go ahead, Dan. For me, I had an epiphany. I went to my dad's little workroom in Sugar Creek, Missouri, and there was a leather-bound book of health in the Depression era, 1920s. And I was about 14 years old, was thumbing through this and saw in there a chapter on consumption. And I thought, oh my gosh, these poor people, nurses and doctors were working at consumption hospitals getting consumption themselves and dying. And I thought, how could they not understand what was going on, with tuberculosis and treatment? But even microbe theory was not recognized, let alone treatment. So you have to kind of forgive ignorance of what you don't know when you look backwards in history. And then if you take the same approach and humbly ask yourself, what are people going to say about what we're doing today, 50 years from now? We're going to look equally as ignorant, perhaps as confused. And with that kind of, gee, I don't know, could we do it better approach? That's how I began my career. Dan, go ahead. And so really simple things for me. When I began my July 1 day as a pediatric resident, I was really terrified because I had seen a senior resident order Valium 0.02531 milligrams for a baby that was seizing. And I thought, how in the world am I ever going to learn that? I realized later, nobody can draw that one up. But I was especially nervous I would miss that septic baby in the ER. And so, 104.5 fever, looks kind of sick. Is it otitis or is it sepsis? Do I do a septic workup? At that time, H flu was killing children. And so I was very worried about how quickly I might learn. And saw a patient I thought was pretty sick, a little baby, and there was a bandemia. So I asked my senior resident, what do you think? Septic workup, maybe meningitis. And he said, it's probably the band lady. So, I didn't know what the band lady was, but being a skeptic, I went up to the reference lab upstairs and, a night on call and found a supervisor, found out there were only a dozen techs, went through all of their data for a 3-month period. And sure enough, there was a band lady who called triple the number of bands over every one of the other 11 technologists. So that's the way my brain was thinking. And if you organize data, ask questions, you're always bound to learn. Just something fun early in my days. I eventually did figure out how to tell a septic versus not septic child, but that's where things started for me, being terrified. On to the next, Dan. I want all of you in the room, and if you're a little bit older than 35, and I'm just slightly above that age these days, I think that ideas are more liberated when you have perhaps less fear and a little bit of a smaller knowledge base. Some of the most incredible things happen for inventions and breakthroughs in that third decade of life. So look what this clever guy did in 1 year's time. All 4 of these could have won Nobel Prize. And do you know which one did? Brownian motion, not relativity, fascinatingly enough. On to the next, Dan. So as I entered my fellowship, I was a young guy, and I was fortunate to move from a good medical school, a good residency, into an extraordinary pediatric hem-onc fellowship, where everything was open to question. Moving the needle on getting children to be cured was remarkable. Tumor boards were a wrestling match. Every single patient was discussed thoughtfully. We were just bursting at the seams with clinical trials, biologic and studies. It was remarkable. I was fortunate to land in such a stimulating place. Dan, go ahead. In my first 3 months on the leukemia service, just for example, presenting grand rounds, a flow of ideas came around how to compartmentalize, organize, simplify, make assessments around things. I saw a patient that was dying on maintenance chemotherapy for ALL after tolerating intense treatment. And finally, I figured out with a Scandinavian lab that this was a homozygous TPMT, thiopurine methyltransferase slow patient. And now Mark Matrana and you folks screened for this enzyme, but dating back in 1985, it was the first patient ever described. And it was curiosity that helped me discover that. Next one, Dan. I had ideas, and I sat in a lecture on an orthopod showing a leg and the various tissues and how a leg, if you have a deep injury, can regrow, not quite to the elegance of a worm that can regrow a whole tail, but I began to think around cell-cell communication and how things work. And it made me really doubt the hematopoiesis slide I had learned in Williams' textbook, where a differentiation occurs and you're inextricably, permanently on your pathway, and there's no other way to make those straight lines go differently. I thought that was just all wrong. I had a very different idea about stem cells and plasticity. I will grant you, once a red cell extrudes its nucleus, it's probably not going backwards ever again. But I think the rules around stem cells later proved very prescient, and probably should have followed my stem cell inclinations earlier on. But nevertheless, ideas were flowing. Next slide, Dan. And after a year and a half in the clinic, I was fortunate to be able to go into an extraordinary laboratory under Chuck Schur, a molecular biologist, head of tumor cell biology, started on projects cloning genes with gene regulation, focusing on my leukemia interests that had evolved by this period of time, and stumbled upon a work trying to understand a hematopoietic growth factor receptor gene, CSF1 receptor, macrophage colony stimulating factor receptor. That was the cellular counterpart for VFIMS, oncogene. And in cloning upstream, very large gene, multiple introns, one very large one before exon 1, stumbled into another gene, the platelet-derived growth factor receptor gene. This was an extraordinary breakthrough for me. Jan, go ahead and go to the next slide. That I was able to capitalize on, and ended up getting a publication as a young guy, still a fellow in Cell. So what a piece of fortune that was around Christmas time. So rather than focusing on the unique nature of these genes, and now sequencing and the anatomy of our genome is very elegantly able to be described, and we can order it on a patient, have results in a couple of weeks. I was encouraged by my PhD boss, follow the science, take this where it leads. You should be an evolutionary biologist. Look at this gene regulation, how a ligand receptor and interactions of turning off and on are operating. And I thought, I don't want to really understand dinosaurs. I want to understand how to cure leukemia. I was not greatly encouraged by my lab boss to follow my inclination into the next pathway. Go ahead, Dan. But I had a question from my literally first month of fellowship: how do we cure childhood leukemia? And so there was a belief that these drugs, much like antibacterials, had activity up to, say, 2 logs, maybe even 3 logs of cell kill, but not much more than that. And go ahead into the next slide, Dan, when you expose patients on maintenance chemotherapy for a couple of years with ALL, the fundamental premise that I was taught was you destroy every single cell, or leaving even one will ultimately result in relapse. And I was struggling with the math of this. A child that's, say, 4 years old has a kilogram of cells. That's 10 to the 12th, a trillion cells. How could 4 drugs get you down to 0? And in fact, if you think of the logarithmic scale, 10 to the 0 isn't 0, it's 1. So go on to the next. I ended up moving to MD Anderson, pursued, and when the technology was available using PCR, a set of probes that were unique to each child with ALL. And a question that I had in my mind for my first month of fellowship, then 12 years later, I think we answered very substantially. Children that are cured in 3 years plus off of treatment in their bone marrows still had leukemia cells related to the original clone and grew in clonogenic assays. It suggests there's something else to cure. Now, you might think, well, that's not surprising. We know the immune system is involved in cancer regulation and control today, but this was dogma bashing back those years ago. So go on to the next. Fresh ideas. My career went where it went. I enjoyed it. I was fortunate. I was surrounded by brilliant people. I stumbled onto a thing or two, but I ultimately was stimulated to help others after feeling I had been greatly assisted in my own career. And here's where I'm going to the next, Dan. I want to share an experience or two of what I believe I've learned from a great teacher. Started at MD Anderson with a wonderful chief medical officer there, Tom Burke, a military guy, extended into my stays in Long Beach and Idaho, how to help others. Go on, Dan. And I really think it starts here, and I'm going to thank my parents and my family members for consciously thinking about leadership choices and principles that are fundamental to your being. And for me, they involve integrity, honesty, being mission oriented, science and medicine as noble pursuits, and working with others. While it's fun to do things on your own, you can get a lot more accomplished when you collaborate. Those are my philosophical principles that I have not ever shirked from. Go on, Dan, to the next. And so for me, I hope this resonates with you in your careers as you consider administrative leadership positions. Way back in medical school, I thought, you know, a year three picks on a year two, a resident picks on an intern. I decided to not foster that. I was going to kind of break that cycle, maybe a pyramid underneath me, encourage those underneath you. Don't even think about them underneath you. Work with people, help each other, see if you can climb the ladder by helping somebody up that same ladder. It's been something I believe since I was a young guy, and it's a key to my leadership principles. Next, Dan, please. And so I want to ask you to think about when you're working with your colleagues as a leader, whether administrative, scientist, or whatever your capacity and role is at Ochsner, think about how you can pull somebody up, like in that chutes and ladders game in the middle, up that ladder, up to the top, so everybody wins. And don't make the mistake of competing, going around, maybe torpedoing somebody else. Do you think that's benefiting me? I'm going to rise. I'm going to climb that step ladder by kicking somebody else down, bringing everybody else up with you. That's my theory of relativity that I would ask you to embrace. It's been how I've modeled and my leadership and my intentions. And I hope in my time with all of you in 4.5 years, I've been able to help in some regard, because that's been my goal. And to the last, Dan. So thank you all for what you've done. I think in my 10 minutes here with you, I wanted to let you know how much I respect and appreciate and admire you guys. Sorry I couldn't be there in person. I will be there on April 24th. I really hope to have hugs and handshakes and great wishes both directions. So thank you all. Thanks, Dan, for the time. And I do wish you a wonderful holiday weekend. Dan, back to you.
Thank you, Mark. That was a really great 10-minute summary of like all the cool stuff that you've done, some good advice. I want to thank you very much for the past 4 years of your leadership here. You've really helped us develop into something good and something that's going to advance to become something extremely special. And you've been a good friend to me as well, so I really appreciate that, Mark.
Thank you, Dan.
In our last few minutes, I'm going to respect everybody's time, get you out of here, back to your families. I wanted to give you an update about our residual fund use. So as you know, we made a policy change 2 years ago in an attempt to try to take advantage of the residual funds that have amassed over time and were sitting fallow, basically, in just accounts and were not being used to advance research further. So what then wound up happening, as you'll remember, is those monies have been gathered together and redistributed in a partially socialist way and partially capitalist way to the various centers of excellence over the past 2 years. And I wanted to give you an update about how the various centers of excellence have used those funds, so that you guys can also think about it and speak with your heads of centers of excellence if you have other ideas about how funds might be used next year, for example. So in cardiovascular, we established a competitive seed grant program. We called it Invest CBD. And in 2002 and in 2003, we have funded seed grants for 2 projects each year. Hopefully those will go on to lead to externally funded research endeavors. Ed McCool in the multi-specialty, both medicine and surgical multi-specialty department, in an extremely original way, made the Invest MS program, which also funded 6 selected pilot projects over the years. Now with William Davis being named the research medical director for the medical multi-specialty group, that group will divide into 2 next year. Transplant developed a pilot project for monitoring pre-liver transplant patients. Hem-Onc bought equipment in 2022 for the Baton Rouge Biobank, and in 2023, they're supporting an investigator-initiated trial examining neurotoxicity. Ms. Rahi is the PI on that. Neuroscience, they were a little more socialist, I guess, distributed the funds evenly to the PIs who had been productive in 2022. And in 2023, they adopted the competitive award process, and somehow were able to award 10 separate grants to 10 separate investigator-initiated projects that will be completed over the course of this year. Remember, those funds disappear on December 31st, so they must be used during this calendar year or else they go away. Maternal and fetal health also similarly put together a competitive seed grant program. I don't have any further details other than that. Anybody have any questions about the residual funds?
I got a grant in October, and then the funds vanished away at the end of the year, whereby I hadn't even gone through the IRS. So there's sort of process of, even if you decide to announce it in January or February, you have to take into account that the project may not be done till the end of the year. It's a real waste of talent for physicians to spend their time and energy to try to create a project that is actually useful for the organization and find out that the funds are not there. Because these are seed funds, you're supposed to collect the pilot data and get onto a bigger grant. That's the idea. So I don't know what needs to happen, but I think, we need to think of strategies of how we're going to get the initial investment to work out for us, our department. Because once the project stops, we're not moving forward. Now we've invested in something that is not giving us any fruit.
I agree with that.
Hey, Dan, Dan, would you mind giving a very brief summary of the comment?
Yes, so that was Salima Kamruddin, who mentioned that she received one of our competitive seed grants. That was the first one. So remember, she was given that grant in September or October or something like that, and then wasn't able to turn around the research in time and wound up losing the funds, and so wasn't able to complete the research that she had won the grant for. And her point was, to paraphrase, that we need more time and we need a different mechanism so that people who win these awards are able to complete their project. Is that fair to say?
And Dan, if you don't mind, I would just say our very first year, we had the choice of implementing nothing or implementing something rushed that was like in the last quarter, and certainly was very, very challenging. Sorry about that, Salima. And the goal is to have everyone have a full year to expend the funds going forward. I'm sorry, Dan, go back to you, my friend.
I totally, so I agree with that. It was extremely rushed that year. We tried to make that clear, but we knew it was going to be challenging for everything. And, we are learning and we're going forward. This year, for example, in cardiovascular, we're going to make an announcement of the availability of the grants earlier in the year, so people can start putting their brains together and their proposals together, such that when we know what our budget is, we'll be able to quickly receive those grant applications and be able to turn around decisions so that people can hit the ground running really very well on January 1st.
A lot of us use it sometimes to start internal projects. So I think it's very productive that we would engage other young upcoming physicians in a program like that and give them support. At the same time, I don't know, it depends on how big the pool is, but for individual researchers who've been doing this for a long time and may want to expand their wings and do another project, I think there should be some kind of an allocation. Those just being pooled in one center. I think some researchers may feel that they do not have any residual funds that they can start, especially if they work for a substantially long amount of time. I'm not one of those yet, but this is something that came to me as an individual. I think research is already so hard as a clinician scientist. We put our blood and sweat. So when we obtain the residual funds, part of it is the hope that you'd be able to use that to support other. Now it's obviously code for our department purposes. So I think there should be some rules around that as to how to actually also be able to support physicians who are maybe have our large residual funders or have done or have a substantial history of making impact in the department. That was something that came to my head, and I don't know if it's right or not, but I thought that I'd bring it up.
I think it's a reasonable concern. I was not part of the decision-making group that changed the way that residual funds were going to be handled. But I did hear that the rationale behind it was so that we could use the residual funds that were otherwise not being used in order to sort of prompt researchers to advance investigator-initiated trials, going forward. Is this the absolute best way to do it? Probably not. But, it at least has resulted in what you see on the screen, and which is a number of different investigator-initiated trials that did receive seed grants, and hopefully will be tweaked going forward. But I absolutely agree with you that if there is any way that we can make it a longer period than 1 year by whatever, accounting processes need to happen, we should absolutely do that, I think.
And I don't want to throw cold water on it, but there is an accounting requirement. The monies have to be used in the same operating year. That's not going to be flexible. It's just the way these board designated funds work. So I think we're going to have to just optimize and make our process as efficient as possible and think of these as 1 year seed funds.
We'll do the best we can. Thank you both. We're heading towards the end here. So as you guys remember, those of you who are old enough, 3 years ago, Bridget, when she was in my position, put together something that she called the Pulse Survey. She took the pulse of all of the physician investigators, or principal investigators in our center, to try to get an idea of opinions and recommendations about going forward and how we should improve things within our research endeavors. As you know, cardiologists like cute names for studies. I'm a cardiologist. So, Bridget had the Pulse Survey. I'm going to have the Repulse Survey. And it's not my favorite. It's not so cute, but I thought I figured I'd give that to you. We're going to do the same exact survey, first of all, as what she did before, so that we can compare the results, over the years and see if we have improved and see where we haven't improved. But I'll probably add some other questions as well based on our developments in the interim. So you should be looking for that in the coming months. Our last slide is the next PRC meeting. I looked forward, approximately 3 months or so to see when would be a reasonable time to do it. And we will hold it, barring any horrible conflict, on Wednesday, July 31st. I want to thank you all for coming. If there's any other pressing questions, or if you want to, shake Leo's hands or get his autograph, you can come up to the front. Have a great evening.
Thanks, Dan. Have a good evening all.
Thank you.
Thank you.